Hello, my name is Ian Fish. The title of my talk today is Frequent Interserotypic Recombination of Foot and Mouth Disease Virus During Subclinical Co-infection with Serotype O and A viruses. But first, I'd like to thank everyone in the ARS lab, especially Dr. Stenfeld, who headed up these animal experiments, and the De Los Santos lab, who helped with the in vitro part of this work. Thank you, and everyone else here and at Plum Island. All right, so as you heard from Dr. Stenfeld earlier, we performed a set of experiments in which cattle were infected first with serotype A virus, and then either 21 or 35 days later infected with serotype O virus. The data that I'll be covering are all from this persistent phase here, and these are oropharyngeal fluid samples, and we Illumina sequenced them. Spoiler alert, we found recombinant viruses resulting from these serial infections. First, I'll provide a basic description of our initial findings. And second, confirmation of these guys and their characteristics. Then some follow-up laboratory analyses that we did with one of the recombinants. And finally, the conclusions. So across those 12 cattle in this arm of the study, recombinants were found at the consensus level in 20 samples from these 5 guys. And 4 of them in at least 3 different samples from different sampling days, and these are 3 or 4 days apart each sampling, we found recombinants at the consensus level, meaning they were the dominant virus that were in those samples. So in order to confirm that these were real, we repeated sequencing and found the same results. And we also regularly found the same recombinant in serial samples, like one sample after another from the same animal. Now, during this talk, I'll have a few alignments that look like this one. And the light gray parts of the alignments will indicate an O-derived part of the sequence, something that looks just like it's O parental inoculum virus. And then the darker black parts that kind of look like barcodes here, those will be the parts of the genome that are derived from the A parental virus. So for example, the first 2 sequences in this alignment are from animal 1914 on days 35 and 38. And they, from 5' through about here in 2C, look like the O parent virus. And then right from early in 2C through the end of the virus, 3', they look just like their A parent. So we further confirmed these recombinants through RT-PCR using primers that crossed the breakpoints, followed by Sanger sequencing. And all the breakpoints matched the NGS consensus level recombinants that were identified. In addition to finding the same recombinant in serial samples within individual animals, we found that also 3 of the 5 animals with recombinants had different dominant recombinants on different days, in at least one clear instance of a recombinant resulting from multiple recombination events, which is pretty cool. The recombinants weren't the only viruses present in most of these samples. So these graphs show the proportions of non-recombinant A to non-recombinant O to recombinant viruses that were detected. So for example, in animal 1930 over here, on day 31, it was about half and half O wild type to recombinant viruses detected in that OPF sample. And then in the next sampling date, it was mostly recombinant with a little bit of A. And then those guys switched places, and then they switched places again, and then the A ended up winning out. And it looked like in most of the cases, the A's usually ended up being the only virus that was detected by the final sampling dates in the animals. Because we did deep sequencing of these samples, we could also use this data to get an idea of any minority viruses that might be present. We used haplotype reconstruction programs, which showed us that, especially in the earlier samples, there were usually other recombinants at low levels. So in this example, from animal 1929 at 31 days. At about 84%, you found the dominant recombinant with a breakpoint around 3A, right here. And then at around 12 or 13%, there was a non-recombinant O virus present. And then at about 3 or 3.5%, there was this minority virus that had a recombinant breakpoint in the middle 3D right around here. And what's extra super cool about finding this guy was that if you took this recombinant and this recombinant and you recombine those, you would get that weird triple looking recombinant you were looking at earlier. So, which became dominant forever in that animal, which was, a really cool finding, I think, here. Another way we looked at this was through looking at chimeric reads, and these are reads that looked half like A and half like O. And those guys supported the same exact breakpoints and the same minority recombinants that we found. And as a side note of interest, in the dual infected vesicles from the simultaneous AO co-infection arm of the study, there was no evidence like this of recombinant viruses. Looking at the overall genomics of the recombinants, the breakpoint locations, you can see that they're all located in non-structural coding regions. All the recombinants encode O capsids, which was the second virus, which kind of makes sense from an immunological point of view. There were no neutralizing antibodies against the O, but there were plenty of them from the A that these animals were infected with a few weeks before. All the breakpoints, all the genomes were in frame without any weird indels or anything. And then we wanted to also correlate the breakpoints with sequence homology between A and O to see if that was important. And then also with broader field studies, studies that looked at lots of other FMDs and see FMDVs to see where they recombined. And we found positive correlations in both of those cases, especially when we included all the recombinants, the dominant and the minorities in relation to looking at all FMDVs that have recombined historically, I guess. Next, we did some follow-up work, in vitro work with one of the recombinants, this 35-day recombinant from animal 1914 with a breakpoint right in 2C. And we plaque purified this guy along with both of the inoculum viruses and with the A virus that ended up outcompeting and beating out this recombinant in this animal. So we compared their plaque morphology, growth in cell culture, and specific infectivities. Plaque morphology analysis indicated that the capsid probably has more influence on plaque sizes than do non-structural proteins. You can see the recombinant looked quite a bit more like the O than the A. And then looking at just regular growth in cell culture, the growths weren't that much different really at all between the different viruses, so we don't predict any loss of infectivity either. The last assay we did was to run a sucrose gradient to get an idea of whether packaging of this recombinant genome might be impaired. And because we found a lower viral particle to PFU ratio, we concluded that packaging of this chimeric half A, half O kind of genome into a regular old O capsid was probably not an issue at all. In conclusion, we found that interserotypic recombination is very common in the nasopharynx following this kind of FMDV superinfection in carrier cattle. And through about 10-20 days after superinfection, the second infection, the recombinants were often found to be the dominant virus. And that these recombinants always, in these cases anyway, seem to encode the capsids derived from the newer virus. It should be the one that wasn't under this, immune pressure of the first virus. And our final thoughts are that, this question, could superinfection of carriers be a viable origin of the recombinant FMD viruses that we identify in the field? Thank you, everybody.
Hello, everybody. Buenos dias, Buenos Aires. It's a pleasure for me to be here virtually sharing with you some of our results on FMD and its interplay with MDA5. Triggering of the antiviral response relies on recognition of viral molecular patterns associated with viral infection by viral sensors. On the other hand, viruses and FMD in particular have evolved a lot of mechanisms to counteract that host response. So, Riga-like receptors are a family of viral sensors, which are cytosolic and ubiquitous RNA helicases. The family consists of three members: Riga, MD5, and GP2. And type 1 induction, interferon induction during the coronavirus and FMD infections are believed to be linked to MD5. However, there are some reports suggesting the involvement to some extent of Riga, at least in early steps of the coronavirus infection. So we plan to address these two questions. Does FMD RNA interact with MDA5, and does Riga play a role? So for that, we performed RNA pull-down experiments, which consist of transfection of flag-tagged versions of MD5, Riga, or to negative control, lacking all the helicases domains. And 24 hours later, we infected those cells with FMDB. And 5 hours after infection, we lysed the cells, analyzed the proteins by immunoblot, and also extracted the RNA associated to each of these IP fractions for RT-PCR analysis. In the immunoblot analysis, we saw that all the proteins, the bait proteins immunoprecipitated as expected. And in the case of MD5, we detected an extra band, which might be the sign of cleavage event going on. Okay. When we analyzed the RNA fractions by RT-PCR using primers annealing either at the 5' end or the 3' end of the FMD genome, we could readily amplify these two regions from the RNA fractions isolated from from MD5 IP fractions, but not associated with Riga or 2-CAR IP fractions. This suggested that the FMD genome was interacted selectively with MD5 and not that much with Riga. Next, we wanted to learn whether the RNA derived from FMD infection had indeed an MD5 dependent immunostimulatory activity. First, we checked that RNA extracted from FMD infected cells had immunostimulatory activity, and that was the case compared with RNA from mock infected cells. And then we infected this RNA or from infected or mock infected cells together with MDA5, Riga, or an empty vector. And as you see here, at 2 hours after transfection, the only significant increase in interferon beta mRNA induction occurred when MD5 was being co-expressed with co-transfected with RNA from FMD infected cells. In this case, this is the result of the input RNA transfected, as there's no sign of cleavage for EIF4G, which is very early upon infection. And however, at 6 hours post-transfection, when this CAP translation initiation factor is already being cleared, so which is an indication of viral replication. We saw the same effect, but with slightly higher level. To close this cycle, we confirm that the RNA extracted from the MDA5 IP fractions had indeed MDA5 dependent interferon beta mRNA stimulatory effect. Next, we wanted to test the effect of RIG-I and the MD5 expression on FMDB growth. For that, we did transfection infection experiments in two different swine kidney epithelial cells. These first SK6 cells are interferon competent, but IVRS2 cells are not able to respond with interferon signaling route upon infection or transfection. So, as you see here, in cells expressing MDA5, there was a 2-log reduction in viral titers, unlike with RIG-I, which basically overlapped with the kinetics of the empty vector. In the case of interferon incompetent cells, the 4 kinetic curves basically cluster as one. So, in agreement with these results, we only detected interferon beta mRNA by RT-PCR or antiviral activity by interferon bioassay in SK6 cells expressing MDA5. These results suggest that inhibition exerted by MDA5 is clearly linked to interferon induction, and that RIG-I apparently had no significant effect on FMDB infection. In 2018, we reported that LGP2 was being another, if you remember, member of the RIG-I-like receptor family, was being cleared by the FMDB leader protease. And we were able to precisely map the sequence, the target sequence for a probe on LGP2, and correlated this clearance event with impairment of the antiviral response. So we wonder if ELPRO might be targeting other RNAs, especially when we saw that this motif, this conserved RNA helicase motif 6 was absolutely conserved also in MD5, and not that much in RIG-I, where there were two substitution in this region. So when we check what happened in co-transfection of RIG-I with increasing amounts of LV, catalytically active LV, we didn't see any sign of cleavage other than the expected reduction in the protein levels due to the effect of LV on translation. And we found the same results under the context of infection. However, when we analyzed the effect on MDA5, we clearly observed two N and C-terminal cleavage fragments during infection, and also MDA5 degradation in an LB dose dependent manner when we co-expressed it with increasing amounts of the catalytically active beta proteins. As we did for LGP2, we generated a triple mutant MDA5 in which these 3 positively charged arginine residues were substituted for negatively charged 3-glutamic acid residues. This triple mutant was completely resistant to cleavage, both during FMDB infection or in co-expression with increasing amounts of of catalytically active LB proteins. And last, we wanted to analyze if MD5 cleavage was impacting somehow on the antiviral response exerted by the helicase. And for that, we took advantage of the cleavage-resistant MD5 triple mutant, which interestingly preserved for antiviral activity. You see here, its effect on FMDB growth was basically the same as the wild-type protein. We did again this transfection and infection experiments and checked interferon induction, viral titers, and antiviral activity. And we show that only when the wild-type versions of both proteins were co-expressed, LB and MDA5, a significant interferon beta reduction in interferon beta induction was observed, unlike in the presence of any of the mutant proteins, MDA5, or the catalytically inactive mutants of LB, or obviously both. So, in agreement with that, we detected the reduction of viral levels in inverse correlation with those observed with interferon beta, and same for antiviral activity. You see here, this mark that when the antiviral activity, which is completely suppressed in the presence of LB wild type, is recovered. To some extent when the cleavage resistant version of MDA5 is present. So today there's an increasing list of proteins which are being described as LPRO targets. Some of them are mapped and share these partly or in a greater or lesser extent this motif, and some others are also very relevant proteins in antiviral immunity, but the cleavage site has not been precisely mapped. So as conclusions, take home messages, just saying that MDA5 senses and interacts with FMDB RNA. Both MDA5 and pt2 are key helicases for mounting the antiviral response against FMDB, and LPRO cleaves them both, taking advantage of conserved motifs in RNA-binding proteins in what we call an all-in-one strategy to impair the RLR signaling pathway at the earliest steps of infection. The identification of this target motif is helping to identify novel LPRO substrates. And just to finish with a very general conclusion, which is that it's quite obvious that the lessons learned from viral pathogens may help us in the development of new strategies against infectious diseases. Just slide, so mentioning people who did and collaborated with this work, and special thanks to Miguel Rodriguez Pulido, who did most of the work I mentioned, Encarnación Martínez Salas and Francisco Sobrino, our collaborators at the Icahn School of Medicine at Mount Sinai, in academia, and our funders. So thank you very much for your attention, and thanks a lot to the organizers for their effort and for the opportunity to share this data with you. Thank you very much.
My name is Lydia Dykes, and today I will tell you how mutagenesis mapping led to discovery of novel functional RNA structures localized within the genome of FMDV. FMDV genomes aren't linear, and several RNA structures have been well characterized for the untranslated regions, such as the S fragment, pseudonodes, CRE, IRES, or SL1 and SL2, all playing a role in replication of FMDV. The structures localized within the open reading frames are less characterized. In 2001, 25 conserved RNA structures were predicted within the open reading frame of FMDV, however, their function remains unknown. We investigated whether RNA structure localized within the open reading frame are important for virus replication. However, since the RNA structure prediction carried out in 2001 was based only on 9 FMDV sequences, we first revisited the bioinformatic prediction using 118 FMDV full genome sequences representing currently known FMDV genomic variability among all 7 serotypes. Here's the map of highly conserved RNA structures common for all 7 FMDV serotypes. The X and Y axes represent genomic positions on a single genome. Each dot marks a single pairing formed by 2 nucleotides, one located at the X-axis and another at the Y-axis. A group of multiple dots clustered together indicate a single stem loop. You can see that we correctly predicted structures localized within the UTRs, verifying that our analysis performed well. Shape of these structures drawn from this prediction can be seen on the right. Looking at the open reading frame, 3 conserved stem loops were localized within the region encoding the L protease. Several serotype-specific conserved stem loops were found in the region encoding the capsid proteins. However, only 4 of these were conserved in all 7 serotypes. 46 highly conserved RNA structures were found in the region encoding the non-structural proteins. Overall, 53 structures, which were conserved among all 7 serotypes, were found within the open reading frame, and on average, these structures were conserved in 112 of 118 FMDV isolates tested. To assess function of highly conserved RNA structures within the open reading frame of FMDV, we employed a subgenomic replicon in which the genomic region encoding the capsid proteins was replaced by a reporter gene encoding green fluorescent protein GFP. Usage of FMDV subgenomic replicon allowed for real-time monitoring of replication by amplification of GFP signal due to replicon's translation and replication. To control for background GFP signal, we also included a replicon with an inactive polymerase, marked here GNN, which is unable to replicate and which gives GFP signal solely due to translation. GFP signal can be monitored over time, showing replication kinetics, and since in our study the peak of replication was at 8 hours, for simplicity I will show data for that time point only. The fact that FMDV's subgenomic replicon is replication competent suggests that there are no RNA structures present in the region encoding the capsid proteins, which are essential for translation and/or replication of the FMDV genome. This is why we focused our study on the region encoding non-structural proteins. We artificially divided the non-structural encoding region into 9 fragments using convenient restriction sites and permuted each fragment using CDLA algorithm, which preserves the amino acid coding and the nucleotide frequencies. The CDLA permutation changed around 10% of nucleotide identity within each fragment, and computational analysis confirmed that the RNA structures localized within each fragment was drastically altered by the algorithm. Each single CDLA permuted fragment was inserted into the FMDV replicon, giving rise to 9 mutant replicons, each containing a single CDLA fragment. Replication of each mutant replicon was tested in 2 cell lines known to support FMDV replication: BHK cells, which are routinely used to grow FMDV for vaccine production, and MDBK cells, which are of bovine origin. The replication kinetic of mutant replicons was compared to the wild-type replicon, shown in orange, and the replicon with an inactive polymerase, GNN, shown in gray. Surprisingly, in both cell lines tested, mutation of RNA structure within 8 regions spanning 2A through to most of 3D polymerase had no effect on replication. Contrastingly, mutation of the RNA structure within the region encoding the terminal part of the 3D polymerase, shown here in red, led to abortion of replication with GFP signal comparable to the replicon with an inactive polymerase. This suggests that the genomic region encoding the terminal part of the 3D polymerase contains functional RNA elements. We investigated the RNA structure localized within the region encoding the terminal part of the 3D polymerase in more detail. The 5 stem loops localized in this region, called open reading frame stem loop 49 to 53, showed high conservation of nucleotide pairing, even higher than SL1 and SL2 localized in the 3' UTR. A part of conservation of nucleotide identity, highlighted here in red, these structures also showed evidence of covariance when nucleotide pairings were preserved despite presence of nucleotide substitutions among FMDV isolates, here highlighted in shades of yellow, green and blue. SL49 to 51 also showed high nucleotide identity conservation at the terminal loop of the RNA structure. To test which of these stem loops are involved in translation and/or replication of FMDV RNA, we mutated each individual stem loop in such way that the neighboring stem loops were not affected. Mutated stem loops were incorporated into FMDV replicon, giving rise to 5 different replicons, each bearing a single mutated stem loop. Replication of these replicons was again compared to the wild-type and GNN replicon in the 2 cell lines as before. Mutation of SL49 and 50, shown here in black, had no effect on replicon replication, showing similar replication profile to the wild-type replicon. Replication of SL51 mutant was slightly but significantly reduced in BHK cells and even more reduced in bovine cell line. The SL52 mutant showed a significantly reduced replication in both cell lines, with replication levels in bovine cells resembling the GNN replicon. While in BHK cells, the SL53 mutant showed replication levels similar to the wild-type replicon, in the bovine cells its replication was significantly reduced. These results show that the 3 stem loops SL51, 52 and 53 are important for translation and/or replication of FMDV RNA, with the SL52 possibly being the predominant structure. The effect of this stem loop mutation was more pronounced in the bovine cell line. BHK cells are known to lack the fully functional interferon pathway, while MDBK cells have been shown to secrete large amounts of type 1 interferons. It is possible that these 3 stem loops, SL51, 52 and 53, might also assist in evasion of the immune recognition. To conclude, we have generated a comprehensive map of the highly conserved RNA structure within the genome of FMDV and identified 3 stem loops which are important for translation and/or replication of viral RNA. Similarly to the viral genome, replication of an FMDV replicon involves viral protein synthesis and the sequential synthesis of the complementary and genomic viral RNA. Further studies are required to dissect which of these processes are dependent on the 3 functional stem loops. It was puzzling to observe that the 43 out of 46 highly conserved RNA structures located within the region encoding the non-structural proteins appear dispensable for replicon replication. It is possible that some of these apparently non-functional structures play a role in viral genome packaging or are genome-scale ordered RNA structures, GORs, which may play a role in persistence of FMDV in its natural host. These functions would be not detected by our replication model system. Although by its limitation, This approach might be used to study factors which can attenuate a wide range of FMDV strains. I would like to thank people involved in this project, the funding agencies, and you for your attention. Thank you.